Hello everyone. Today we are going to learn about difference between simple staining and gram staining. So moving forward, coming to our first aspect of this differentiation is purpose. So in what purpose, for which purpose actually we use simple staining and gram staining. Okay. So here for simple staining, it is commonly used technique to visualize the morphology of the bacteria. Okay, so the simple staining, this is a technique which is commonly used for the morphological study of the bacteria. Now, which are the characteristics that comes under morphology? Okay, so there are three types of morphological studies. First one is shape, second is size, and the third one is arrangements. Okay, so all these shape, size, and arrangements, this topic has already been covered in our previous video. You can go and watch it. Okay, if you want to know in details. Now, here in shape, as we can see, there is a bacillus, coccus, viridis, spirillum. Okay, now there are different types of sizes also present. Zero point 0.1 micrometer to 2 centimeter, and arrangements, there are different types of arrangements as well. Okay, so all these morphological characteristics can be studied using simple staining. Whereas in gram staining, this gram staining is a type of differential staining method, right? So it differentiates bacteria into gram positive and gram negative based on cell wall composition. Okay, so the cell wall composition here, it is a structural visualization. So this gram staining helps us to do the structural visualization of the bacteria, right? So, based on the cell wall composition, it divides into gram positive and gram negative. So, in our previous video, we have also covered their cell wall structure. Here, the structure is shown here. This is the gram positive cell wall structure, and this is the gram negative cell wall structure. Okay, so based on this structure, the gram staining helps to differentiate between the gram positive and gram negative bacteria. Okay. Now, coming to the simple staining again, it does not differentiate between cell types. Okay, so it will not help you to differentiate between different types of cell types as because only one type of stain is used. That's why. Whereas in gram staining, more than one type of stains are used, and therefore it helps in identifying the bacterial species very clearly. Okay, it helps in separation of this bacteria into different groups. Okay, so these are the purposes of using simple staining and gram staining. Now coming to a second aspect. Second is where reagents use. So what are the reagents that are needed to carry out these experiments? So for simple staining, from the name we can already understand that the reagents or the procedure, it might be simple as well. Okay, so here comes the reagents that are used. So here in this staining, you can use either positively charged dye or negatively charged dye. Okay, there is no need to use two types of dyes, only one type of dye. Okay, either positively charged or basic dyes or acidic dyes and distilled water. Only two reagents are used. Whereas in gram staining, there is a need of using more than one types of stain. Okay, for differentiating the groups of bacteria, more than one type of stain is used. So here the primary stain, crystal violet, is used. Grams iodine is used as mordant. For decolorizing agent, 95% ethanol is used. Sometimes acetone is also used, and a counter staining stain is also used, that is saffron. Okay, so here we can see more than one types of stains are used for gram staining, right? Now, coming to the procedure. Okay, so this is the pictorial flowchart of the simple staining. So, first, you make a smear on the glass slide. Now, remember, we have already talked in our previous staining videos that before making the smear, you need to wash your slide first, make it grease free, so that when you make a smear, the smear doesn't get damaged or distorted the shape. um, you need to have a proper shape to visualize in the microscope. Okay, so this will allow you to get a proper shape. Now you make a smear, you air dry it, and then you do the heat fixing using a Bunsen burner, and after that you put a positively charged dye like this blue color dye, which is methylene blue, or negatively charged dye. Here it is Congo red. Okay, so you can do this staining by using either positively charged dye or negatively charged dye. Okay, and then allow the stain to interact for 1 minute on the slide. Okay, and then you wash it off with distilled water, and then you do the air drying. You dry your slide, and then you observe under the microscope. You can observe it at 40x or at 100x also. Okay, so this is the simple staining procedure. Now coming to the gram staining procedure. Okay, so we'll have a quick review. Now here you make the smear, and then you air dry it, and after that you do the heat fixing. Now before heat fixing you need to air dry your smear. Okay, now this point is important. Then after that you add the primary stain to the smear, which is crystal violet, and you keep it for 1 minute, and then after that you wash it with distilled water. After distilled water you add iodine, which will act as a mordant, and you allow it to react for 30 seconds. Now this mordant will help the crystal violet to interact properly with the smear, with the bacteria, with the bacterial cell wall. Okay, and it will make a crystal violet and crystal violet iodine complex. Okay, so this acts as a mordant. It allows the interaction of the stain with the structure, with the cell wall of the bacteria. Okay, then after this 30 seconds you decolorize the slide. Now this slide, if it has the gram positive bacteria, they will already retain the crystal violet color, and those that are not gram positive, they will remove their CV when decolorized with the ethanol. Okay, so then after that you add safranin, which is a counter stain, and you keep it for 1 minute, and then you wash it with distilled water again. Now after washing you air dry it and observe under the microscope. Again you can see in 40x or 100x. Okay, now coming to next aspects, that is the result. So what can we expect in the slide when we look it under the microscope? Okay, so in the simple staining, if we use the positively charged dye or basic dyes, so here methylene blue is used, suppose. So here you will see that all the bacteria has been colored in blue. Okay, so here in this picture you can see there is a coccus present, there are some rod shaped bacteria present, some cylindrical shaped bacteria present as well. Okay, all of them have stained blue color. So all the stains will uniformly stained with only one single dye color. Okay, now if you have taken any negative stain or acidic stain, here suppose Congo red is used, so here you will observe somewhat this type of image. Okay, so here you can see that the background is stained red, while the bacteria have not taken up the color and they are appearing uh, clear. Okay, they are appearing clear here. So here also you can see the morphology in both the staining, in both the simple positive staining and simple negative staining, you can clearly visualize the morphological structures. Whether it is a coccus or rod shaped, you can clearly visualize it. However, you cannot see their structural um, composition. Okay, you cannot differentiate them based on their structural composition. However, in gram staining, there is a gram positive bacteria. So in gram positive bacteria, they will retain the CV color, that is the crystal violet, and will appear purple in color, somewhat like this. Whereas the gram negative bacteria, they will retain the safranin and they will appear pink in color, somewhat like this. Okay. And there are other two groups which are also present. One is gram variable. They will retain the stain unevenly and appear as a mixture, somewhat like this. Okay, so some will take up the saffron in color and some will take up the crystal violet. So you can see mixture of bacteria are present. Now, in gram non reactive, they will retain the color very poorly, or sometimes they do not even retain the color, and they will um, be seen somewhat like this. Okay, so this gram staining, it is differentiating the bacteria into different groups. It is separating the bacteria based on the structural composition into different groups. Okay, so here we can clearly see the difference of using the simple stain and the differential stain, that is the gram staining. Okay. Now coming to the mechanism. Now what is actually the reason behind this type of staining? So first, in the simple staining, if you see, it uses the single dye. Okay, either positive or negative. So since it uses a single dye, it stains clearly. It stains all the bacterial cells the same color. Okay, all the bacterial cells will stain same color. Whereas in gram staining, more than one type of dye is used. Primary stain is used, secondary stain is used, and again modern is used for proper interaction. So because of the usage of more than one type of stain, we can see different types of bacteria having retained different dye colors. Okay, so this is the difference in mechanism. So here, if we in the simple stain, if you see the positively charged dye, what they do, they bind with the negatively charged cell components of the bacteria. Now the bacterial cell wall is actually negatively charged. Okay, this also there are nucleic acid which are negatively charged because they have phosphate groups. Now the cell wall is negatively charged. So whenever we use a positively charged dye, what it does, it interacts with the negatively charged components and they retain the color properly. Okay. Whereas the negatively charged dye, they stains the background as the cell wall repels the negatively charged dye. Okay, so there's a theory from the physics. We have already heard about it. I think you all have heard about it, that the like charges repel each other and unlike charges attract each other. Okay, so here the positive charged dye will bind with the negatively charged cell wall, whereas the negatively charged dye will get repelled by the negatively charged cell wall. That's why they will only color the background. Okay, so this is why the morphological structure is clearly visible. Now, an example of the negatively charged dye is given in the negative staining. Okay, this negative staining is also covered in our negative staining video. The link is in the description. You can also go and check it out. Now, in the gram staining, what happens? The gram positive bacteria, they retains the crystal violet color. Why? Because in the gram positive bacteria, they have a very thick peptidoglycan. Right? They have a very thick peptidoglycan and their lipid content is very low. Okay, so therefore there is the interconnection is very high because of the thick peptidoglycan, and the crystal violet does not come out of the cell wall. That's why it retains the crystal violet. Whereas the gram negative bacteria, they retain the saffron in color because of their higher lipid content and thin peptidoglycan. Now gram negative bacteria also have an outer membrane which is not present in the gram positive bacteria. Okay, so this is the mechanism behind the retain of the different colors of the dye. Okay, now in the simple staining, we have seen that the stain, whichever stain you are using, the stain uniformly colors all bacterial cells, irrespective of the type or cell wall structure. So in the result section, we have already seen whether it is coccus or bacillus or spirillus or vibrio, whatever it is, it will have the same color of the dye. Whereas in gram staining, they will show different types of colors presenting different types of bacterial groups. Okay. So here in the gram staining, iodine, as I said, it acts as a modern, plays a very important role in the gram staining because it increases the interaction of the cell components with the dye by making CV iodine complex. Okay, so this is the mechanism of simple staining and gram staining. The detailed mechanism of the gram staining is covered in our gram staining video. If you want to understand the detailed mechanism of gram staining, you can check out the link given in the description. Okay. Now coming to our next aspects, that is the applications. So where these applications, the simple staining and gram stainings are used. Now the simple staining, as we have learned, it is a very quick and easy method to observe the bacterial morphology, right? Now it is used in teaching labs or when details of the cell walls are not required. Suppose we just want to see the morphology only, we don't want to get into the details of the cell wall. So for that purpose, simple staining is very much accurate method to do this. Okay, preferred method. Whereas in gram staining, this is essential in diagnostic microbiology to differentiate and identify bacteria. Now, since gram staining is a type of differential staining, so it is used in the diagnostic microbiology. Okay, for differentiation and identification of bacteria. Now, it is also used in pharmaceutical companies. It is very much useful for creating antibiotics, since gram positive and gram negative bacteria respond differently to the treatments. Okay, so to understand the working mechanism of gram positive and gram negative bacteria, and to prepare their specific antibiotics, we need to understand their cell structure. And first, we need to group them as gram positive and gram negative, which is given by gram staining. Okay, so gram staining helps us to understand the cell wall structure of this bacteria properly. Okay, so these are the applications that we have for simple staining and gram staining. Now, coming to the summary. Okay, summary of simple staining versus gram staining. So we'll have a quick recap of what we have learned in this video. So here are the aspects. Purpose. So in purpose, what are the purpose that we have studied? For simple staining, it enhances contrast by coloring all cells for observation of shape, size and arrangement. Yes, we have studied that. In gram staining, it differentiates bacteria into gram positive and gram negative based on cell wall structure and composition. Okay, this is clear. Now the principle that is used in simple staining is that it uses a single dye and uniformly colors all the microbial cells. Whereas in gram staining, it is based on the ability of the bacterial cell wall to retain the crystal violet after decolorization with alcohol. Okay, so this is the principle. Now the stains that are used here in simple staining, it is mostly preferred basic dyes. Okay, basic or positively charged dyes are preferred, like methylene blue, crystal violet, saffronine. Okay, so these are the single basic dyes. So only single type of dye is used in simple staining, whereas in gram staining, more than one type of dye is used. So here we can see there is a primary stain, there is a modern use, there is a decolorizer, and there is a counter stain. Now in the decolorization step, for simple staining there is no decolorization required, whereas in gram staining it is an essential step to differentiate between gram positive and gram negative bacteria. Now in the mechanism section, the dye binds to the negatively charged cellular components such as nucleic acids or cell walls. Okay, so this is for simple staining. So in simple staining, we have learned the mechanism that the positively charged dye will bind with the negatively charged cell wall structure. Okay, whereas in gram staining, the crystal violet binds to peptidoglycan and iodine forms a complex. Whereas decolorization removes stain from gram negative cells. Okay, so this is the mechanism. Now coming to the cellular features. Uh, in simple staining, the cellular features, this simple staining does not distinguish between types of bacterial cells. It will just do the morphological analysis. Only general morphology is observed in simple staining. It will not differentiate the types of bacteria. However, gram staining will differentiate the cells based on the thickness of the peptidoglycan and lipid content in their cell walls. Now, what is the outcome from the simple staining? All cells will appear the same color based on the dye that is used. Whereas in gram staining, the gram positive cells will appear purple and the gram negative cells will appear pink or pinkish red. Okay, so this was a quick recap of the comparison between simple staining and gram staining. Uh, I hope you all have understood this video. If you have any queries related to this video, you can put your comments in the comment section below. And if you want a detailed notes of this video, you can follow our LinkedIn page. The link is in the description as well. If you have liked this video, do give a thumbs up, share and subscribe to our channel for more such related microbiological updates. And I'll see you in the next video.